so that we can begin. And also in the meantime, we had sent out a set of 5 questions as a pre-test to this session. I'd request all of you to fill the answers to those questions and submit as soon as possible. Once the session is over, we'll be also sending a set of questions as post-test.
So would you like them to upload it before we start?
Yes, ma'am.
Yeah.
We have received 25 answers so far.
Okay.
A very good afternoon, all our dear students. So we all know that research it has gained a lot of importance in the last couple of decades or so, and in all fields, including medicine. And the speed at which advances are being made in medicine and the strides that are being made in research, we have to keep ourselves updated, and we also need to participate, because unless we do that, we may end up lagging behind. So when it comes to biomedical research, Ramya Medical College has always tried to promote an environment which is conducive to perform high quality research. And we know that we have a lot of potential. We have the infrastructure, we have the support in terms of the Division of Research and Patents, which is always ready to guide students, as well as a number of faculty who are willing to be guides for students in their projects. But the main asset that we have is our students, who have always displayed a lot of enthusiasm when it has come to performing research. And we know that there is no research training that is included as part of MBBS curriculum, but as an initiative from our college, under the guidance of Shalini Nui ma'am, and via our Division of Research and Patents, our head Nankumar sir, we have decided to conduct these sessions in order to try and help our students to get acclimatized to how research can be performed, and right from the basics and on to a slightly complicated or slightly advanced sessions in research also. So today's session, we plan to have it as a sensitization session, just a sort of an icebreaker, just so that you can have an exposure to what basically goes through in performing research, in biomedical research at undergraduate level. So I request Shalini ma'am, our vice principal, to say a few words before we start with the first talk.
Thank you, Dr. Akshay. I'm really glad, and I'm really happy to see so many students have logged in. But it would be really nice if I can see your faces. Would you mind putting on your videos, please? All the students, can you put on your videos, please?
Could we request everyone to turn on the cameras, please? Go ahead, turn on your cameras.
Put on your cameras. Neha, Mohana, Yashaswini, Manvita, Hasita, Isha. It would be nice to see you while we're speaking. And I'm glad some of you are multitasking. You're doing your exercise also while listening to our research talk. All right. Next time onwards, please be ready to put on your videos, okay? It'll be good to see faces while we are speaking instead of looking at a blank screen. Okay. So at first, what I thought of is it is such a lovely and initiative by the medical college and the Division of Research and Patents that we are encouraging undergraduate students to take up research. Uh, Even if you don't take it up, at least to know about research and to know what it is really, really important. Although research, as Dr. Akshay said, is not a mandate, it is not part of your curriculum. We still talk to you about it because we want you to be updated. We want you to be among the best in the country. Most, if you look at, the very prestigious medical colleges like Postgraduate Institute, Chandigarh, All India Institute of Medical Sciences, Vellore, what is that, CMC Vellore, Manipal, all these prestigious colleges encourage research, not only among faculty, among postgraduates, but also among students. It is important because you get to know the practical aspect of things. You get to know how exactly, a new idea takes shape, how exactly we can give evidence of the benefit of a new idea. And it has also been found that it helps you to become better physicians. It helps you to become a better doctor and to give better care to your patients. Okay. So although it is voluntary, I think it's a very good step that you all have taken to listen to what is research. And probably many of you will take up research, which I am 100% sure will be very beneficial in your careers. And now more and more institutions are making it mandatory for you to publish papers in journals if you want to advance your careers. Particularly Western colleges, if any of you want to go there, you would have to have good research experience to get admission in good medical institutions. All right. So I'm really happy that our faculty are, taking the initiative, spending so much of their time to help you conduct research. So please take this guidance positively and conduct some small research, which will be very helpful to you. And all the best to everybody. Thank you, Dr. Akshay.
Thank you very much, ma'am, for those encouraging words. So students, please, as ma'am said, make full use of these sessions. I know many of you are currently doing projects or wish to start projects now. So you will have a lot of doubts in your mind. So this is a good opportunity for you to be able to clarify any of these doubts that you have. So in case you do wish to ask a question, once the talks begin, you can add those questions in the chat box, or at the end of the sessions, all the talks, we could have a question and answer session also, where you can unmute and ask your questions directly. So as part of the session, the first talk is by Dr. Tanuja. She is associate professor in the Department of Ophthalmology, and the topic would be formulating a research question and a hypothesis. So I request Tanuja ma'am to begin her talk.
Thank you, Dr. Akshay. Can you see my slides?
Yes, ma'am, it's visible.
Yeah, thank you. A very good evening to all the students. So I'm Dr. Tanuja, Associate Professor in Ophthalmology. But as a member of DRP, I'm be handling this topic of formulating a research question and a hypothesis. Now, before we go to this, I want to tell you a story. Okay, this maybe some of you have already heard it. Some of you have not heard it, so it'll be good. But for me, when I first heard this story, it was very inspiring, and it captures the whole essence of. Sorry, it's not moving. Just a minute. Capsulates the essence of research. Just a minute. Just give me a minute. This is not moving. Can you see my this one now? Can you see my slide now?
It's the title slide, formulating a research question and a hypothesis.
Oh, okay. Sorry, no, that's not the one.
You want to put up another PPT, is it?
Yeah, no, the PPT was not moving. I'll just do this.
Okay.
Can you see it now?
Not
Sorry,
yet.
not yet.
yet.
Hmm.
Yes.
Now
Story time.
Can you?
Yes,
Yeah,
story time.
So this was a very inspiring story when I first heard it, and I want to share it with you people. So there was this Irish surgeon who was working in Uganda in 1957. Okay, so he was working in his OPD, and he used to see these students. I mean, not students. He used to see these patients, young African individuals who would come with swellings of the neck, swelling involving the buccal mucosa, and to his surprise, they were always having a very high mortality. Now, this raised the question in him, and why was it happening, and what was it? So he asked around with other of his colleagues were they seeing the same thing, and all of them said yes, they were seeing, and they all said they have very bad mortality, so leave it. So, but this didn't stop this Irish gentleman surgeon. He started doing a research. I mean, he started studying what the literature of what was there. Was it reported earlier? And to his surprise, in 1901, somebody had, published something in a tropical medicine journal saying that this kind of tumors were quite common in African countries, and the pathologists had reported it to be a sarcoma. So this surgeon wanted to publish it, and he quickly wrote up a manuscript titled "Sarcoma Involving the Jaws in African Children." I say manuscript because it never got published and he never got a reply to it. But again, it didn't stop him because he was continuously seeing these patients and he wanted to do something for it. He had the drive to do something. He wanted to do something for it. So what he did was, he over 4 years he sent out to these questions. He sent out posts to people all around Africa, asking them whether they were seeing the similar kind of disease in their geographical area. And after for a long 4 years, he got 400 responses, and he presented these. He presented these findings in a European conference in Middlesex. So what he did again was he put pins on the African map and found that there was a geographical distribution for this. It was definitely not there in South Africa, not there in few countries up north, but in the middle of Africa, it was there. So in that particular conference, there was a pathologist, two pathologists, Tony Epstein and UN Barr, who got interested with this. And they were like, okay, when they again did a geographical biopsy, they went back, collected the biopsies of all these, of whomever they could. And they found, and they also did the demographical study of these patients and found that this disease, this tumor was not affecting the brothers and sisters of the patients. It was not affecting the siblings. But it was very unique that it was not like a genetic, but it had a geographical distribution. So they started doing reviewing the biopsies again and found that it was not a sarcoma. In fact, it was a lymphoma. And then they thought, okay, since it did not have any genetic, this one, it was unlike any other cancers which was presenting. They thought, could it be an infectious agent causing a cancer? Which was unheard of till then. And that's when they found that something was very typical, very similar in all these things. but they couldn't identify an infectious agent. So what they thought was, can we find antibodies? Do we find antibodies against a particular virus? And it clicked, and they found that there were antibodies to a particle in all these patients. And that's how they detected the first virus, which all of you might be aware of, Epstein-Barr virus. And that's how they found that Burkitt's lymphoma. Burkitt is the surgeon who was doing all these collecting the information, collecting data and everything. Burkitt's lymphoma was associated with Epstein-Barr virus. And meanwhile, Burkitt was not sitting alone. I mean, he was not just saying, okay, somebody else will do the study and find out the cause. He was trying treatments. He was trying what was the treatments for this, and he tried methotrexate, he tried cyclophosphamide, and luckily for him, he found that it was all getting treated and it was getting cured. So in this, what you see is, this is the story of how within a decade, I say 10 years, a cancer was discovered. So it was not a sarcoma, it was a lymphoma. The morphological fingerprint was identified, geography was delineated, and a causative agent was discovered, and a treatment was started. Now, this is an impressive story during 1950s and 60s, where epidemiology was not well developed, was not well defined. And with all its constraints, this man, Parsons Burkitt, did it with his grit, with his perseverance. And the most important thing for him was he allowed his mind to ask this question, why was it happening? And he wanted to pursue it, and he achieved it. So where do you get these research ideas? You get research ideas from seeing patients. You're seeing patients and you don't know what is happening. You get research ideas when you read certain literature, when you talk, when you discuss with your colleagues, you attend conferences. The most important thing for research to happen is this question, why. You don't let it die. You know, you need to keep asking. You see something abnormal, you keep asking this why, and you pursue on it and persevere with it. And that's when research originates, and that's when we find new things in medicine and probably find treatments for diseases which we are always think have high mortality or are affecting the human beings. So in this, we'll take this as a hypothetical situation. If you were there in his situation, in Burkitt's situation, so you were seeing a series of young patients in Africa coming up with similar neck swelling and died. So what would you do? I mean, this is just an hypothetical situation that I'm giving, but for you working now onwards, seeing patients, there might be many situations where you'll come up with ideas, but you need to give it a life and then take it through and give it an end. And that is what is research. So if you were this person Burkitt, you would be probably like, what are the demographics? Why is it affecting only children? Or, is it affecting your elderly also? Is it affecting only in a particular area? What are the symptoms and are there any complications? I mean, how fast does it progress? What is the pathology of it? Is it infectious? That was the last thing that occurred to him only when he presented in some conference and somebody else put an idea, okay, could it be an infectious, So you basically need to be thinking about some problem, and then you will find a solution to it. So a research idea is an uncertainty that starts in your mind. You're seeing something, there's a problem that you identified, and to make it deliberate and meaningful, you probably have a hypothesis. You go to the next stage of an hypothesis where you say, okay, I am seeing it in young individuals. So is it more common? This lymphoma is more common in young individuals, young African and adults? Young African individuals. So that's a hypothesis. So to make it a research question, is it more common in young individuals? That becomes a research question. So in other words, a research question actually details the problem statement. It further describes and refines the issues under study. It adds focus to the problem statement. You have a problem which is an uncertain idea. It has evolved into a research question, but it gives you a direction. It helps you in collecting data and analyzing it and sets the context of research. So research question is the first thing which starts in your study, and it's very important to get it right. And you're not in the era of 1950s and 60s where you have one whole decade to study. And if I tell you you have to do a research for 10 years, I don't think so any of you will be interested. You want everything fast and you want it to happen. So for that, a well-defined research question is very important. So in other words, in epidemiologists and statisticians language, research question is very important. It gives meaning to your research idea, and it is the beginning of your well-planned study. Okay. So research questions can be of two types, like your studies itself. So when we come to research studies, they're either descriptive or analytical. So even research questions can be either descriptive or analytical types. So if you if it is descriptive, it involves observations or to measure or the quantity. Like, you want to describe it is more common in children, it's more common in this particular demographical distribution. So that comes descriptive. Whereas analytical, you want to compare groups and interventions. Compare if you find more antiviral antibodies in the patients affected with lymphoma compared to those without. That becomes an analytical study. So again, here, if we go back to the series of young patients in Africa, that was the research idea. So what are the things that you can think of? What are the studies that you can think of in this? It's like, what is the prevalence of this tumor in a particular area, in a particular geography? What is the pathology? What are the clinical features and mortality and, incidence? What is the causative agent and what is the treatment? And if you have a treatment, is this treatment more effective than something else? Or is this treatment even effective? These are the studies that you can undertake, isn't it? So to begin with, a research question has to be specific that one's audience can easily understand without the need for additional explanation. You can't say, I will do a study describing the whole demographics, I will identify the pathology, I will do the treatment, the study about the treatment also. That becomes too, it becomes lost. It is too general and you will not have a focus to do it. It should be focused. When I say narrow enough, if you're going to do a study only on a descriptive study, it has to be specific to that. You're not going to say, I will also do analytical, I'll do a combination of both, and then I'll make make number of papers out of it. No, one study you can make multiple papers, but if you have your research question itself very broad, you will get lost and you will not see the end of that study. It has to be concise, expressed in fewest possible words. And when we say that, again, it has to be complex also. It shouldn't be like an yes or no question. Is it more common? Everybody knows it is common in this one. Is that just enough? No, it has to require synthesis and analysis of ideas and sources prior to composition of an answer. It also has to be open to debate. It shouldn't be like you're just proving some facts that has already been present. Then nobody is interested in such kind of research. So how to develop a research question? So you have a broad idea, divide that question into concepts. So you can have multiple research studies, but divide it into each concept. Specify the population. So this is a mnemonic, PICO, we say. The P for population. Specify the population in whom you're going to study. What is the intervention, if there's any intervention that you're going to do? Comparison, is there a control group and a comparison group? And what is the outcome? What do you want to see in there? This has to be defined and be there in your research question before you start the study. So how do you arrive at the research question? First is you need to review. So you have a research idea. You need to review the state of art information. You need to do a literature survey and see what all you can what has already been studied, what is already there. If you're going to study the same things, then nobody will be interested in it. So, and if it is already proven, dusted, then there's no point in researching the same things again and again. Then you raise a question. In this particular case, we will see, okay, we reviewed the literature on similar tumors and found that is this tumor more common in a particular geographic location and also which age you want to study the demographic patterns or you want to do a descriptive study. Is it worth investigating? This particular disease had a very high mortality and it was worth investigating. Sometimes which is already proven, it's already a fact, it's well established. And if you want to investigate, if you want to go do research on that, It's a waste of money and resources, and it's usually not worthwhile doing that. You need to next define the measurable exposures and outcomes. So here, I will start developing a research question. Are children in a particular area, or probably in Uganda, in Africa, susceptible to this disease? So that becomes like, I'm measuring the prevalence, and I'm also defining what is I'm going to see in the children, and the outcome is whether they have lymphoma. What is the etiological agent? That becomes a second study. You can do so many studies out of it. Do they have certain antibodies to the virus as compared to those who did not have the disease? Then once you define the measurable and exposure and outcomes, you sharpen the initial question. So now this looks, there are so many questions. I don't want to go into everything, but I will probably limit myself to antiviral antibodies. So I will say, to evaluate the presence of antiviral antibodies in patients with lymphoma compared to those with no lymphoma. Now, already here, it is becoming more focused, and I know that I'm going to be only studying a certain thing, and I'm not going to get deviated and get lost in while doing the study. Finally, refining the question to estimate the amount of antiviral antibodies in young children with lymphoma as compared to children without lymphoma. So what started as a research idea now has become a research question, and with being more focused, clear and concise, and is giving me a direction of what I want to do, and also give a value for the resources, where my resources will be used. So once you develop a question, this is the question that I've developed to estimate the amount of antiviral antibodies in young children with lymphoma as compared to children without lymphoma in Uganda, Africa. So there is a population, there is a intervention. I'm going to study the antiviral antibodies. There is a outcome, and everything is defined, and in this particular research question. The most important thing that you need to do before you go ahead with this research study is this study going to be, can you conduct this study? For that, we have something called a mnemonic, which is called as finer maps. So basically, is this feasible? Can I do it in my particular setup? Okay. So this person, Burkitt, started in 1957. He didn't have any funds. So he wanted funds to do it. So he kept on presenting in European countries and finally got a grant. And that's when it was feasible for him to do this study. Before that, he was only doing a descriptive study. He was just presenting, collecting data. Okay, this many number of children are affected, but he couldn't do antiviral antibodies. He needed funding for that. And for that, so is it feasible to do in your study? Sometimes what will happen, you start the study, and midway you'll realize, oh, okay, this is not really possible. Then probably you need to write down why it was not possible, so it will help you when you plan other studies. Is it interesting? Now, something which is already known to everybody, it is not really interesting, and no, the medical fraternity will not really be interested in your research. So it has to be interesting. Is it novel? Now, when I say novel, you don't have to think that you have to identify a virus, new virus, and name it your name, either Harshita or Neha's virus or anything like that. No, it didn't have to be so novel, but it has to be novel in a way that it shouldn't, whatever is expressed, whatever is there, you have to look it into in a different way. So something which is already, again, established fact, nobody is interested in knowing that whether it is there or not. It has to be something interesting and novel for people to even listen to your study. Is it ethical? The most important thing, you have to do ethical practices. You can't just take people, participants, without their consent. You have to follow all the ethical practices and then only conduct a study. Is it relevant to the particular? If you are just studying some gene which is not even, some disease which is so rare, which does not affect anybody, and it is not relevant to the current population. So again, nobody will be interested in your study. So it has to be relevant to the present situation. Is it manageable? Manageable is almost similar to feasible. When we say manageable, is it manageable in your particular setup is what we're thinking. It might be feasible in a larger setup, but in your setup, and when you are doing that research study, can you do it, is called what is something which is called as manageable. Is it appropriate? Any study you do, it has to be publishable. You don't do some study and just keep all the data somewhere and keep it to yourself. You have to let people know. So again, that becomes very important. And whether you've done it in a systematic way, whether a research question which is very properly described will give you a way to go forward with the research, doing the study. So characteristics of a good research question, which I've already said earlier, is it details the problem statement, it describes and refines the issue under study, adds focus to the problem statement, guides your data collection and analysis so that you're not lost and you're collecting data for everything, and sets context of the research. Now, once we have defined the research, though we started with the research idea and we had an hypothesis, but when you're doing a study, you first tell the research question and then you go on to describe the hypothesis. It's a specific version of a research question. It's usually stated only for analytical questions. For descriptive studies, you don't really have to state hypothesis. For descriptive studies, you are going to see what is the prevalence of the disease or anything like that. You're not going to state a hypothesis that for descriptive study, it is 30% of the young children are affected. No, that you cannot do because you are going to study it. But in analytical questions, you can say, okay, I'm going to use methotrexate. Methotrexate works well in these patients with lymphoma compared to cyclophosphamide or compared to something else. So that's when your hypothesis comes. So, and the main elements of hypothesis is you have to have a sample on whom you're going to do, what is it you're going to put, and an outcome. Exposure and outcomes have to be described in the hypothesis. So the characteristics of hypothesis is it has to be simple, specific, it has to be specific, and you have to have individual hypotheses. You can have many hypotheses in a study, but you shouldn't try to include everything in one hypothesis. And it has to be stated in advance so that you don't get again distracted while you're doing the study and provides you with a focus. So when we say that, so possible hypothesis in this case, so in this particular example that we've taken is lymphoma is more common in Africa compared to Europe. So I'm comparing it. I'm analyzing between one continent and another. So go to study a group of individuals in Africa, a group of individuals in Europe, and see what is the prevalence of lymphoma in there. Or lymphoma patients with high antiviral antibodies respond to methotrexate. So again, it's an analytical study. So you're already stating it, and your whole research study will be to prove or disprove it. So again, like your research question, even a hypothesis has to have a population and intervention, exposure, and an outcome, and also a dose relationship or time relationship. So here I would be writing as children below the age of 16 years, population in Uganda with lymphoma have a high antiviral antibodies, have a better response to methotrexate. So an intervention and an outcome when given within 30 days of symptoms. So here it becomes more specific and it is guiding us on how to do our research study. But what happens with stating it much earlier? You are biased. You will not look into anything else. Okay. You will not probably look into anything else that can be happening, and you are more focused on identifying only these antiviral antibodies, and you might forget that there's some complication happening because of methotrexate. So one may ignore other outcomes which may be possible in the study. And again, as I already told, descriptive studies, hypothesis cannot be stated, and you have to go around investigating and finding out what is the outcome of the study. Now, once you have the research question, once you have the hypothesis, you need to state the objectives, and these have to be framed in scientific and epidemiological terms. So take the question in a few limited axes. Again, don't try to include everything in one objective. You can have multiple objectives and make use of no more than one verb for each objective. When I say that, you are not going to say to evaluate and study the prevalence of of something, So you have to use only one verb. And again, to study is a very bad verb. You don't use that. In case of descriptive, you will probably use the term to describe. Okay. In case of analytical, probably estimate, to estimate the prevalence of lymphoma in Africa compared to Europe, something like that. Okay. So here again, to estimate the prevalence of lymphoma in African children. So these become the objectives. Though when we started with the research idea, when I was talking in the beginning, These were all there in your mind, but now it is becoming more systematic, and we are giving them a meaning, and writing them in the correct epidemiological terms. So how good is your, to end with, how good is your research question? You've written everything, you think you've done a fantastic job. Ask yourself the question and try to answer it. Sometimes the research question you have written, I mean, the answer you might get also might be of no use to anyone, or there might be no answer at all, and then the whole exercise of your research study becomes futile. So try to reframe it. And if your question is wrong, then there's again, you wasted your time and energy, resources, everything on a study which you will not get any outcome. So the question has to be right for you to move forward. That's it. And what I would say, like to tell all of you is, research is very gratifying. I mean, when you see your paper, when you do a study research and you get some results, or when you publish it, it gives you a kind of, it gives you a gratification that is not achieved doing anything else. You do get, as doctors, I mean, as students, all of you aspire to be very good doctors. The gratification that you see when you see your patient happy, is definitely worth for, doing medicine and doing all these hard studies of so many hours of study. But when you do something systematic, do a research, publish it, and it gets published, that's a different kind of happiness. And I hope all of you, aim for it and get it and experience that joy. That's it from me. Any questions?
Thank you very much, Tanuja ma'am. It was a very lucid talk. Tanuja ma'am is a tremendous teacher, and she has given a lot of tips in this area, which is very important for students. A lot of students, when they want to do research, the first step is to form or formulate a research question, and I think this talk has helped a lot of our students to uh, get guidance on how they can go about in order to formulate a research question before they begin the research projects. So thank you very much, ma'am, for the talk. Next topic is will be a talk on research, will be a talk on data collection and analysis by Chidambaram Murthy sir. Sir is the principal scientist and part of the Division of Research and Patents. Sir has a phenomenal number of publications and high expertise in the area of research. So again, I request students to make full use of the session, not just now, even later on, because sir is very friendly and very welcoming. Whenever anybody has doubts, right from undergraduates to senior faculty, he's always welcoming, and he ends up clarifying all our doubts in regards to research. So I request sir to begin his talk.
Dr. Akshay and Dr. Chidambaramurthy, I may have to leave in between,
Sure.
but I'll try to log in from my phone.
Sure. Yes, ma'am.
Thanks a lot for
Thank you, ma'am.
everything. Dr. Nandakumar.
Yeah. Thank you, ma'am.
Thank you.
Thank you. Hope you can all see the slides.
Yes, sir, it's visible. It's visible.
Okay. Yeah. Good evening, students. In continuation of where Dr. Tanuja left, I would like to give a brief introduction as told by my colleague, Dr. Akshay, who is the coordinator of the program. This is a session where we are trying to introduce you to some new terminologies and just give a brief of tomorrow when somebody talks, you should be aware of these terms, and before even you get started with your research planning and execution. So what I thought was today I'll just give perspective for those who are not met me. I'm Dr. Chidambaram Murthy, who is not a medical doctor. I'm a researcher, I'm a pharmacist by training and PhD in biotechnology. So I just want to give a perspective like non-medical and other health science related perspective and what is the research in simple terms, because most of the time research is considered as one of the serious issue and it is not a cup of everybody's thing. And we always feel that it is something to do career. Research is something to do the career. But my personal belief is that, especially in medical profession, research helps you to be a better professional, and also in day-to-day life, it helps a lot to instead of seeing things, you start observing. So that's what the research culture brings you in. And then why? As a student, why should I start research? Like we complete my course and then I start. What are the advantage of initiating or taking up the research as a student? And then the cycle of research, where how to proceed. Should I stop the research after completion of my studies and how to go about it? And coming to the main topic, what are the means of collection of data or information? And I'll give some brief information on how to analyze and how to organize your data for analysis. So let us not go in detail today. The next 10 to 12 minutes, I'll be telling on this and how to represent your data. Once you collect the information and analyze, the next thing is you need to either use it in practice or put it in a public platform for others to use it. So that's what a publication or representation is all about. And how to represent, and we'll summarize that. And as I told, see, this is what a simple research is. You can see two prisoners were asked to write what they see through window. So one person wrote what he is seeing immediately, all the metal grills, what he see. Another person wrote the beautiful nature, what you can see through the window. So this is the difference if you become a research-based doctor and otherwise a regular practicing doctor. You'll be doing still doing your duty, but your vision and focus and all your target will be very close by, and you miss the bigger picture. So research is how you observe things differently from others and put it for perspective which can help mankind. So basically, you try to observe certain things in your day-to-day life or in your laboratory or in the patient and try to come up with something that can help other patients or individuals. And it also depends on the quality of research, depends on the depth of knowledge. So basically, the prerequisite is your interest as well as your detailed knowledge in the area, which will take you to the next level of research application. Yeah, this is something which I thought I will brief for the benefit of all of you. So why we need now it has been we are a little early in introducing it to you, but from next year onwards, like once they start this new education policy, it say 50 to 55% of medical or any professional education should be research based. So whatever we were studying so far on a textbook based or existing knowledge will be soon made into a research based knowledge. So it is more like most of you heard about the activity based learning. When we were kids, we used to do a lot of activity through which we used to learn things. Similarly, doing the research will teach you non textbook information that is essential for both your profession as well as your life. So it helps to understand the concept and its application. Otherwise, you'll be reading and imagining with our limited limitation with imagination. Sometimes we just accept the things without understanding. But involving the research will provide you a better insight on such ideas, and it also best way to integrate knowledge from different background. For example, once you become a doctor, you never think like an engineer or a mathematician or a chemist. So when you are doing research, it makes you think in all angles, and over a period of time, you make the habit that okay, this is the perspective. So it will basically broaden your understanding on the subject, and always it encourages the interdisciplinary learning, something which is, for example, some of you might already know that music therapy, the person who is good in music can bring that to a healing therapy or treatment, and yoga therapy, alternative medicine, all these things. If you have fair knowledge, it's not only to recommend to your patient, you can also follow it for better quality of life. So that is the advantage of this. And it also gives a professional confidence, like once you know the subject in depth, and also you have your own hands-on experience, so you can justify your profession very well. And it also, this is by one of the dean from Australian University. She mentioned that it always helps in developing a long-lasting habit and critical thinking, which is essential in today's medical practice. And in addition to this, it will also provide a better opportunity to serve, as Dr. Tanuja was explaining, how that lymphoma was discovered. So if he had not made a consistent effort, probably would have not had a good treatment option for that. Each researcher, for that matter, everybody's effort has culminated into today's medicine practice. So that is the advantage. And if you are aspirant of postgraduate and higher education globally, if you want to compete with within India, and even if you want to go outside India, research is an important criteria for you to obtain a postgraduate seat or postgraduate course admission. So with all these things, personally, I would say it will make you a creator of some information than a follower. Most of the time, if you just know the treatment pattern, we just follow that and proceed. But if you want to be a creator, definitely it will help you. You can create certain evidence, and you will be having your name forever in the area of medicine practice. So these are some of the advantages with this. This is a typical research cycle. We start with the top 12 o'clock position, read and as Dr. Tanuja told, read to identify a topic. I know a lot of undergraduate students came to the division having many good ideas. So what I tell them is just read more information, update whatever is available. The idea will be very good, whatever you have. See the feasibility, read and identify a topic. And once you read and identify, probably you need to, as a student, you need to initially have a mentor to say that your idea is feasible and it can be taken into next stage, and then do a research under the guidance of a mentor. And the next cycle is publish. Once you do, either you present or publish, so that it will be known to rest of the world, and follow your publication. So initially you start with one, and then try to go deeper in that area, and do more advanced research, and then slowly you start collaborating, and then again the cycle never stops there. If you are a serious researcher, I know the busy clinician of the city is still involved in 2 to 3 major research. They never stop research, because they feel that they learn more and more. And once you publish and present, again you identify a newer topic and continue to do advanced research and publish. Once you make it a habit, it will be real fun. As Dr. Tanuja told, it will have a lot of positive effect on your personal life and as well as professional life. And yeah, these are various schemes. We start from student, even if you become a faculty or a practitioner. So every stage you can get involved in research, and there are a lot of funds available. And probably in future sessions, we'll all take you through the opportunities and other things. So this slide is to show that the research, if you start it early as a student, you can always advance in the cycle, and you can get early benefit of research outcome. And these are some of the things which we'll be teaching you in later thing. And this is something specific to all of you. As a student, you identify a major issue. Two students came and told, like, sir, the food we eat and the health status. It's a fantastic area of research needed for today's generation. And then you try to understand what are the types of food we eat, what is making the change. The food is a very complex system. I'll just go with that example. So it need not be one thing. We know we have plenty of food varieties, starting from junk food, organic food, and I told them understand the background. Why? What is the key component that may be causing the issue, and then to work with that, identify a mentor who has a better knowledge in that area and prior expertise, so that you can get your research started quickly and come up with some successful story, and then discuss the issue with your mentor, and also if possible with group of people, because if you discuss with multiple people, probably epidemiologist, a lab scientist, or clinician, medicine expert, or if you want to see the impact on oncology, discuss with multiple people and collect their information and their opinion, and you can then formulate into a small concept with the feasibility, and then the cycle follows. Like what Dr. Tanuja told, the same cycle. You have to define the problem and design a research experiment, and how to collect the data, like what type of data will help you to say that this hypothesis is proven. So then what type of sample you're working with? Are you working with the questionnaire to identify the issue? Are you working with specific sample to understand the biochemistry, pathology, or molecular biology in the disease? And once you do that, analyze the data and you publish. Again, this cycle is a standard cycle, whether you're a student or a faculty. So if you get used to this in an early stage, probably you can work on a specific topic for longer duration and gain more confidence and output. So these are some of the advantages. So once you publish, probably you can also reach a stage of a discovery, patent, and process, which nowadays the students are starting from high school level. They are getting exposed to research, and they do a lot of innovation, not in biomedical or other field also. So this is the culture we are trying to introduce. Yeah. So this is one slide where what are the advantages, you being a student of Ramaya or Ramayana, what are the advantages? We have a lot of prerequisites that are required to get the resources, both in terms of facility as well as the financial support from government and international and national agencies. We have a certification or eligibility. The Rama institution has established all these things from last 10 to 12 years. So we have successfully completed many student projects. ICMR knows what Ramaya student can do and achieve. So these are some of the existing advantages. And we also have a lot of centers. I want all the students to visit whenever you are free, understand what is this, each of this center of excellence or research center can help you and how they can support you. Once you know the resources, probably that will help you to plan your research. In addition to that, this is the campus where we have almost 21 disciplines within 200 to 300 meter vicinity. So you can access to various multidisciplinary institution as well as the expertise to culminate your idea into research. So these are some of the advantages. And coming to types of research, before I go to data collection, so we have a lab-based research where most of the time we work on the diagnostic lab or discovery lab, the wet lab research. And we also have observational research. So basically, this is more like collecting the existing information and making sense out of it. And then we have a translation research, what we do in lab to be translated to clinical practice. And the clinical research is something directly to deal with the patient and observation. Population based is again the epidemiology and other things. I think that Nandakumar will explain more detail about that. So these are the research which we need to know. And once you initiate your research, then probably in your next stage, once you do one or two projects, you can think of sponsored, competitive, and investigator driven. These are next stage of research. And coming to the major component, before we collecting the data, I want all of you to, as I told, this is only introduction, once you formulate the concept, keeping in mind the outcome, where you want to reach and what is your thing, always try to work on declaring the ownership of the data. I know you are doing the research, and if you're taking help of many faculty or mentors or department, make sure that everybody will get a respectable credit in terms of whatever the outcome you come. Sometimes there are various steps, like you can make them part of the study, you can acknowledge them. So make sure that you thank everyone who helped you in the cycle of research. This is something which we need to keep in mind throughout our life. And then scientific and ethical approval. So we need to subject all this research idea for both scientific validation and ethical approval. Before doing these two, we cannot proceed with any biomedical research. So make in future course, we'll just explain in detail how to get all these things, what are the things involved. And then you have to have a permission as a written document. We call it as informed consent. If you're using a patient information, you have each patient owns his own health data. So we need to take a permission or a consent from them. And if you're using a set of information from particular department or a division, you need to have a written permission before even you access that, and explain them what you are going to do with the data and get the thing. Once you get all these things, rest of the things are in your hand. Like these are the prerequisite before you start any kind of research. And organize your resources. Like as I told, before you plan your research, you should have a certain aim. With sitting with your mentor, you should tell, sir, after doing this research, I know the ultimate outcome is its utility to the public. But before that, I want to present this to this meeting, or I want to publish this. If you have that in mind, it will help you to plan your research in such a way that you don't miss any information for your final outcome. So again, once you start collecting data, so as I told, collection of information, never limit the information detail. So just because you don't have time or other things, so collect as many detail as possible, and you never know, any minor information can lead to a good observation when you analyze the data. So have the broad version of data information, and we have to use all the standard tools to collect this information. And whenever you are doing a lab-based experiment, always you have to stick to a standard protocol. Like we have in vitro IVD approved, NABL and NABH, and we have a standard. And whenever we are recording the information from the patient or patient sample analysis in the clinical study, we need to collect the information with all the consent, as well as the details as much as possible. Don't restrict your, I get, my hypothesis says that it is only age, so don't restrict to age. It may be tomorrow, once you analyze the age, then this present, then they say what is the difference between male and female. Then again, you cannot go back and analyze. So get all the information as much as possible. And again, for both observation and population, you are working with the existing data, but you need a permission to use this. Here, the permission is very important. And again, in continuation of this, you always remember that you are collecting data for the final outcome and presentation. You should know where to, if you have a plan how to analyze that, then it will help you to get the complete detail. And once you complete the, especially in biomedical research, once you complete the study, you will not have access to either patient or any material. So at least in lab, we store for some time and get the access. But in clinical study, once the patient finish the treatment and leave, you may not be able to access them, and they may not give permission or approval to take. So get the information, detailed information at once. And when you're working with the units, in collection of specific data, the unit matter. The unit in which you collect and represent the information should be of international standard. And also, when you're, we may say that nobody will see our raw data. What they see is only final result. But nowadays, for both authenticity and people who want to work in detail, will always request you to show the raw data as an evidence of doing it. And when we use, as I told, if you want to do any measurement yourself or use the measurement, always use a standard thing. Even a diagnostic, there may be inferior diagnostic techniques which may not be acceptable by international things. That then in such cases you may not be able to publish such information. And the survey tools what you use for population-based studies should be optimized, or it should be taken from a good source, WHO and other standard sources. And yeah, these are some of the information, like when you have to record the patient information, whatever maximum information you can take, because you will never know, each one of these may play a critical role when you analyze the final information. So don't restrict the collection of data within the available limit, and whatever the purpose you mentioned to ethical committee, please give a broader perspective and get maximum information that needs to be used for your analysis. Again, yeah, as I told, do not run to a statistician or a mathematician to analyze your result after your collection of the data. We always advise you to decide sitting with the statistician before you start the work, so that they can give what exactly is the sample size, what is the method of analysis and representation, or how to do inference. So most of the time I have seen students complete the study and then they say we would have collected XYZ and you have missed these things. And this simple reason will definitely make you, all your effort go waste, because if you don't have certain parameters, they cannot analyze and you cannot come up with a good inference, and that will not lead to a quality work. So please discuss, plan well. The bottom line is you need to plan well before you even start. And whenever you are selecting a method, please select a method that can primarily associated with your objective. Once you have a hypothesis is a single statement. After that, you split that into one or two objective. Please collect at least 2 or 3 support evidence for each of the objective to justify, and in turn these objectives will address your hypothesis. And again, you have to go for internationally accepted method of both collection and data analysis. And when you are trying to represent the data, please use, we have biological replicates and experimental replicates, probably will explain you later. So you need to have, when you are doing a lab test, at least 3 samples should be analyzed for the concurrence of the result with a duplicate. So use all this standard method. And once you do this, finally, when you are representing data, either in the presentation or in conference, please choose a right method to represent your data. Probably in statistics, will detail with this, like you have graph, either histogram, charts, or 3D pie chart. So you can see these are some of the information which can really confuse when you have multiple sets of information. You are not representing the data to confuse people. It is to convince a certain thing. So make it very clear, and you should know when to use a table, and what images can, sometimes we say image can speak 1,000 words. So what kind of image needs to be included, and the complex things can be made into very simple using flowchart, cartoons, and illustration. Try to use this when you are presenting your outcome or a result appropriately, and that will help. With this, what I would say is start your research very early as a student, and start communicating, not only publication, discussing the research with others, and collaboration, and it will lead to success. With these few words, if you have any doubt, you can just put it in chat box, or you are more than welcome to DRP. We can take questions one-to-one and discuss and help you guys to take up the research. Thank you and happy learning.
Thank you, sir, for discussing on data analysis and data collection and analysis in a very methodical, step-by-step manner. Our next talk would be by the head of Division of Research and Patents, Nandakumar, and that will be about methods. So it's very popular among our students, and he is well known in public health circles, not just locally in our country, in fact, even internationally. So one more reminder that our students must take benefit of the presence of our Division of Research and Patents in order to further their publication research ambitions. So I request Nandakumar sir to speak on the topic of methodology, research methodology.
Thanks, Dr. Akshay, for that kind introduction. And there are around quite a few students who are still brave enough not to log off. So what I'll do is I'll just ask a couple of them. Honestly, in the last half an hour, many terminologies that have been introduced seems alien and also a little bit scary. So we never thought that research will be so complicated and will require us to get so formally trained. Because many of us, especially in the initial phases, we would think that research is something cool and something which we can do it as a routine. But Dr. Tanuja, Dr. Chidambar Murthy are introducing so many complex terminologies and they're saying it has to be done, it has to be internationally accepted. So makes us feel a little bit scary. So I would ask one of the students to just unmute if possible. Riti Shetty, are you there? Riti Shetty? Roshan? Roshan? Ishita Gupta? Guys, if you're none of you are unmuting, then we are assuming that you guys are not there. So you can as well as stop the sessions. We will go home.
Students, this is an interactive session, so please do not hesitate. Sir is
I'm
asking
just
to unmute.
asking you to unmute, guys. Arjun.
Yes.
So can you identify yourself, the person who is talking? Arjun? Ashwin?
Yes, sir.
So can I know who is talking? Okay, this is Ashwin, is it?
Sir,
Okay.
Ashwin talking, sir.
Yeah, Ashwin. Can you just say what we discussed, at least the headings of what we discussed in the last two presentations? We are waiting for you, Ashwin.
Sir, sir, can you hear me, sir?
Yeah, loud and clear.
So first we talked about research, then the research data collection, analysis,
Okay.
and then
Sir, forming a research hypothesis.
Can you please identify yourself, whoever is talking? ARWA is what the name I'm seeing. Can you please tell your name and then talk?
Sir, Arva.
Yeah, please go on, madam.
How to form a research question. Then data collection.
Very good. Okay.
Forming a hypothesis.
Yes. Then why do we need to do research? Brunda H. Brunda H.
Sir.
Yeah, Brunda. Why do we need to do research?
So it'll help us in our professional careers.
Very good. So do you want to do research in your undergraduate days, during your undergraduate days?
Yes, sir.
Okay, so I've noted it down and Akshay will contact you for a topic. Guys, come on, chill. This is like the whole efforts that the Division of Research and our team we are trying to do is to encourage you. And if you guys don't interact, what happens is like the very fact that we are trying to have these interactions beyond the working hours so that like you guys are free, you can develop this aptitude. That is the purpose. Otherwise, we can introduce it as one of the mandatory lectures, have a test for you, and we'll give you a certificate. Good, end of it. So the whole idea that we are trying to give something extra beyond what is uh, mandatorily required is to encourage, motivate, and supplement and provide that kind of an additional support for you to be more capable. If you look at your peers, today it is not a luxury to learn about research, but it is a necessity. As Chidambar Murthy was mentioning, today wherever you apply, half an hour back I had one of your senior friends from the recent batch who's graduating and doing their internship ask for a testimonial with specific emphasis stating that the person has conducted research during their undergraduate days. So be it be USMLE, be it be PLAB or any other exams that you'd be writing and going in for residency or any training, the first thing they will ask is your research acumen. Unless you are able to understand and interpret, even if you are not able to do research, that is a minimal skill that is expected or from your competency based curriculum that you guys will be required to take exam. That is one of the minimal competencies that you would be required to exhibit. So in that context, unless you interact, unless you understand and use the lingo or terminologies of research, it would be difficult. So please, when we ask you to unmute, unlike a second or third grade students who are not able to, in fact, they do a better job. I have kids at home who unmute when you are asked to unmute. It's very demeaning and not certainly not motivating for us as teachers at the end of the day to call out the names and having no response from the other side. So please ensure that there is some kind of an interaction when the senior professor asks you to switch your videos on, or the minimal that you can do if you're on the platform is to unmute and say that, look, I don't know the answer. Nobody is going to hold you responsible for that. The whole idea is to interact, to encourage and motivate you. Okay. So having said that, I'm going to very briefly share the content. 1 second. So can I request Chidambar Murthy to make me the presenter? Please let me know whether you are able to see the screen.
We can see the screen, sir.
Thanks a lot. Thanks a lot. So in the next probably 10 or 15 minutes or much less than that, I'm going to talk about why research methods are important component of the entire research that you would be carrying out. What are the major components in this? What is meant by a data management plan? Chidambar Murthy just spoke about how you collect data, compile data. I will tell you like what needs to be done of it and a little bit about data analysis. So what is research methodology? When we talk about methodology, it is like agar if you want to make 2 cups of tea, how are you going to do it? So the questions that would be asked is whether it is green tea, black tea, tea with milk, hot tea, or maybe cold tea. I don't know, like some people might drink cold tea, not cold coffee. So the questions that would be asked at the beginning is, even before you start doing something, what is the plan with which you will do? So if you know that you have to prepare tea for 2 people, you know the quantity of water that needs to be taken. If it is green tea, you know that like you need to have either the tea powder or dips accordingly, and also with sugar, without sugar. All these things that come as a part of preparing that end outcome as per your objective is what is called as research methodology. It basically answers the question of among whom or who are the people for whom the research is intended, where are you going to do this research, and how you are going to do the research. So it means that it is a blueprint for action, and it provides a guidance for both planning your activities and the resources that would be required, both in terms of human resources, material resources, money as a resource, and time as a resource. So how much time would you be requiring to do this task is what your research methodology is going to talk. And also the most important part, the last one, what I've written as reinventing the right wheel, for the simple reason that most of the times the research falls into 2 broad categories, something which is new and innovative. But most of the times when we are talking about research protocols, we see that it falls into the other category of 3R, unfortunately. Either repetitive, redundant, or regressive. So what do we mean by this? So if somebody else has done it, so you're repeating the same thing. Or it is redundant, it is not of much use to anyone. Or it is sort of like somebody else has done it and you're trying to do it in a different way, which is still okay, provided you're very clear about means to that end. So in that context, when you're talking about the research methodology, you'll be required to talk about these following components. So what are the components? The first and foremost is the study area. So when we mention the study area, broadly it can be categorized as the geographies where the research is happening. And it has a huge bearing for the simple reason, as Dr. Tanuja mentioned in her example, the geographical distribution. So when we are teaching you about epidemiology, we talk in terms of distribution, in terms of time distribution, place distribution, and person distribution. So the place distribution is what refers to as geographical distribution, whether the study is located in an urban area or a rural area, peri-urban area, some locality. For the reason, if you look at the total accidents, so the accidents that can happen in urban areas are different from that on something which happens in a highway vis-a-vis something that happens in a small village. So the mortality might be higher on the highways because the average speed will be higher and most of them result in fatal collisions. As against when you're looking at an accident which happens in a small village road which is bumpy, it might lead to fracture or it might lead to something else. So in that context, mentioning the study area is extremely important. So also, if you're taking a hospital-based study vis-a-vis a community-based study, so total number of deaths due to head injuries at Nimans versus total number of deaths due to head injuries in Jayanagar area or in Bangalore. Which do you think will be higher? In Jayanagar, if you take the total number of deaths, at Nimans, the deaths due to brain or head injuries can be higher. For the reason it is a tertiary care hospital where more severe cases with poorer prognosis can be coming in over there. So in that context, mentioning the study area and choosing the study area for your objective is extremely important. The second thing is the study population. Who are the people whom you would want to focus the study on? Again, this depends on your study objectives. And if you're saying total injuries or the epidemiology of child injuries, we recently had a project from Indian Council of Medical Research, which was funded project, a multi-centric project at 10 different sites with Ramaya as the coordinating site. So the whole idea was to understand where and how children in the age group, let's say 6 months to 19 years are having and what are the different types of injuries what is happening to those children. So in that context, inclusion and exclusion criteria becomes very, very important. And it is a fundamental thing which you should realize as researchers. What do you mean by inclusion and exclusion criteria? It is a simple thing that, like when you are saying epidemiology of childhood injuries among children, so children are your study population, and it can be urban and rural area based on the study area. But you have to say, are you going to take all children or a subgroup of children? If you're taking a subgroup of children, what is the criteria for the child to get included in the study? If the child has any physical deformities or psychological or any mental deformities by which the child is not able to indulge in such activities which might have a bearing on the outcome or the injury, like for road traffic injuries to two-wheeler drivers, unless the person is driving a two-wheeler, they will not be sort of like having that particular exposure. So in such instances, you need to say that whom you would want to include in the study population. But more importantly, among such people whom you have included, now for this epidemiology of childhood injuries, we would have included all children in a particular age. Let's say 6 months and above or 1 year and above, depending on if it is, if you want to exclude the infants, we will have a cutoff age, like let's say 3 years to 19 years. Among such children whom you have included, are you going to exclude any subgroup because they are not likely to have that exposure? So in such cases, the exclusion is a subset or a smaller group among the larger included population. So many times we see that, like if you're doing a study on prevalence of anemia among females, adolescent females. So who are the people whom we are going to include? Can I ask one of you to respond?
Adolescent females.
Very good. So you're going to include adolescent females. Whom you are going to exclude?
Males and non-adolescent females.
Very good. So you're going to exclude males and non-adolescent females. I'm sure you would have heard this joke. All the students who are not in the class, raise your hands. Sounds stupid, right? When I say that. If the students are not in the class, how can they raise their hands? So exclusion criteria is only for those whom you have included. Among adolescent girls whom you have included, if there is any bleeding disorders, if there is any like what you call as morphological defects which might have a bearing on that anemia, then such people who are not likely to be similar to the other group, you are going to exclude because the pathophysiology in that group will be different. So please remember that exclusion criteria is always a subset of only those whom you have included. So if you have included males, the exclusion should also be the males. Because while you are including the other group, like the females are not at all there in the study. So please get it into your head that you can exclude only those whom you have included. So inclusion and exclusion criteria are like subsets. Is that clear?
Yes, sir.
Great. So then we will move on to the methods of data collection. Murthy mentioned that, like, okay, you have to define the variables, what you're going to collect. Let's take a simple study where you're going to measure the growth of physical growth of primary school children in Kaivara. It's a very simple study. The objective is to measure the physical growth of primary school children in Kaivara. Can I ask one of you to tell what you're going to measure for physical growth? Can you please repeat?
So can we measure the mid-arm circumference?
Excellent. You can measure the mid-arm circumference. Then?
Weight, body mass index.
Very good, Ullas. So you're going to measure the body mass index. For that, what you will be requiring to measure body mass index?
Sir, I would require equipment to check the height and weight.
No, I was talking about the variables. Basically, body mass index is a derived variable, which means that if you have
height and weight.
Very good. So if you have the weight and if you have the height, weight in kgs divided by height in meters square is the body mass index. So you will be getting that. So for the sake of simplicity, let us take three things what you are going to measure. One is the mid-arm circumference, height, and weight. So now, one of you, can you tell that what instruments will be required to measure this?
So weighing scale,
Very good. You need to have a weighing scale. Okay.
measuring tape.
Measuring tape. So measuring tape can be used both for the mid-arm circumference and also for measuring the height. Okay.
Yes, sir.
Fine. In that context, when we are referring to the instruments, in the research methodology terminology, the instruments can be either the actual instruments like measuring tape, sphygmomanometer for blood pressure, or they can also refer to the questionnaire or the tools that you can use to get that data. So many a time, so probably in your clinical postings, you would have been exposed where you're going to assess the mental health status, mini mental state examination, there is a questionnaire. Or for quality of life, you have the WHO WHO call. Or in our community medicine, we will teach you for measuring the socio-economic status, Kupaswamy classification or a scale. So all these things which you are going to collect the data is called as instruments. So the terminology here does not only refer to a thermometer or some physical instrument, but also the questionnaires or tools or techniques by which you are going to get the data. While you are doing that, calibration is a very important component, which unless you set the weighing scale to 0 before you measure, if the weighing scale, let's assume that it's an analog weighing scale and the spring is slightly loose and every time it shows 2 or 3 kgs more. So in such cases, unless you calibrate it to 0 or measure that variation from the 0 so that as a systematic error, you can either subtract it from all the weights measured, some kind of equal measurements every time you measure is called as calibration. So that is very, very essential while you are collecting the data. So when if it is an instrument, we call it as validating it or calibrating it. And the last thing is the metrics or the variables that you have to measure. As we told, for the current study, if you are assessing the physical growth of children, we have got three variables. One is the mid-arm circumference, height, and weight. So the metrics is you have to be very clear. You want to measure in centimeters, weight in kgs, and mid-arm circumference in centimeters. For the reason that if you have a tape which has only inches and two people measure or the same person measures it, there can be errors. So the instrument and the metrics of the variables should be very specific. So the last thing that you need to understand in the methodology is your data management plan as to how you will collect the data, what are the variables that you will include in it, and whether you're getting that information. All of you answered this pre-test or some of you have answered. So we had made a questionnaire on the Google form that is an example for a digital, mobile or app-based collection. So you can use a web form, you can use a Google form, you can use mobile apps, tick and click, or any kind of such digital applications for getting the data. Or you can give a questionnaire and ask you to tick and click. So the next thing is once you have collected the data, suppose if it's a paper-based data, you have to enter the data, you have to clean the data for any inconsistencies and prepare it for statistical analysis, which will be either in terms of a descriptive statistics or inferential statistics. So we will talk about these things with examples in the coming weeks. So what we will do is we will give a small example or a study for you to work, and next week or the week after when we again start this interaction, you will get used to using these terminologies and probably start having a protocol which you can do research on. So I will stop here with descriptive and inferential statistics, and I would request Akshay to continue the next session. And if there are any quick doubts or something which you would want to ask me, we will respond. Otherwise, we'll move on with the next session. Any doubts at this junction?
Sir, you talked about inclusion and exclusion criteria.
Mhm.
I quite didn't get that point. You said you should exclude the people you include or something like that.
Okay, so basically, if I say that you go to Starbucks, okay, Varun,
Okay.
say yes here.
Okay. Hello.
Yeah. So you go to Starbucks. There are 6 items on the menu.
Hmm.
Okay. You don't like chocolate.
Okay.
4 of the 6 are caffeinated drinks. I'm talking about beverages.
Hmm.
2 of them are chocolate drinks.
Okay.
Okay. So in the menu, so that is the entire inclusion criteria. So 6 items are there. So that is included. So what you can exclude, since you don't like chocolate, 2 chocolate drinks you will exclude.
Yeah.
And in the remaining 4, you will choose one of them, right? Did you
Yeah.
get that? Suppose suppose you say that I will exclude strawberry milkshake, which is not at all there in the menu.
Yeah. Yeah. Okay.
So
So
that can't
that's the
happen. Got it? So we
Okay.
can exclude only those we have included means actually it is slightly complex than what I'm trying to simplify, because the inclusion and exclusion criteria depend on the universe from where you are going to select your sample.
Okay. Okay. Okay.
So that will depend on the objective that you would have set. Suppose if you want to study something, for that objective, your inclusion and exclusion criteria will align that, so that when you do that research, you will be able to extrapolate the results, because most of the studies will be done on smaller samples. You cannot take the entire population like census, you count everyone. But research studies are always done on a smaller subpopulation. So how do you include and include the population in such a way that the results are representative to the larger population is the whole funda of statistical sampling. So we have not spoken about it. So to ensure that representativeness, you will have the inclusion and exclusion criteria, which will help you in refining the extrapolation of results to the population to whom you are doing the study on. So maybe in the subsequent classes, when we talk about sampling size, sampling methods, probabilistic and non-probabilistic sampling, we will confuse you more and try to clarify it. Is that okay?
Great, sir. Thank you.
Great, great. Okay, so I'll request Dr. Akshay to continue.
Thank you so much, sir. Sir has explained a very vast and complicated topic in a very simple manner. So, but as sir said, we will take this forward in a more hands-on approach in the coming weeks. This is meant to be just a sensitization session. So, I'll move on to the last topic for the day, that is ethics in biomedical research. So we have learned now with regards to research what we want to do, why we want to do, and how we want to do, isn't it? So we're all good to go now that we're clear about what, what, why, and how we want to do the research. We can take it forward, right? So before that, for those who don't know me, I'm Dr. Akshay from Department of General Medicine. So this topic is very important, and it's quite dear to me. So I'll be spending the next 45 to 60 minutes speaking about this, just 145 slides. So because I want to speak, right? So, but is that correct? Is it only important about what I want? No, right? So this is an interaction between a speaker and a listener. So if it's only what the speaker wants that is important, and he doesn't take into calculation or consideration what the listeners want, then it will not be fair, right? So it has to be done, whatever we do, it has to be done whenever there's an interaction, whenever two beings interact, whether it's two humans or whether it's a human and an animal, there has to be empathy. So I just wanted to highlight empathy. No, don't worry, my talk is not going to be anywhere near 45 minutes or 60 minutes. So the topic for today is ethics in biomedical research. So why I want to emphasize on empathy is having empathy goes a long way in trying to figure out where ethically you would be right. So ethics is wherever there's a conflict of interest. So defining ethics is a little difficult, but one of the definitions given is nature of conflicts arising from moral imperatives and how best we may deal with them. So it's not so easy to define ethics. Rather, it is something where when we experience that situation, we get to know. Okay, so there is a situation where there are two options. Which option is better? Neither of them may be wrong, but which one is the better one? So one more way to define or one more way to describe ethics would be it in research. It is the moral budget. So whenever you do a research, you want to do a project, you will have a budget. You will have a financial budget. So you want to have some expenditure in order to perform that particular or execute that particular project. But at the same time, what about the moral budget? That means, is the research being conducted at the expense of moral values, or is the research being conducted at the expense of virtues? Are we ending up uh, discarding human values or humane values in order to perform that research? That is something that we need to keep an account of. So when we talk about ethics, we're trying in biomedical research, we are trying to not just think about what we want to do in terms of the research or why we want to do or how we want to do. We also have to think about the participants' point of view. So now that we are going to be recruiting participants, whether it is a human or sometimes in animal experiments, it could be animals. We need to make sure that we don't infringe on their rights. So we may want them to, for example, undergo a particular test or maybe take a particular drug. But will when we ask them, when we invite them to participate in the study, we should make sure that we do not sacrifice or we do not violate any of their rights. So we must make sure that any research should be done in an ethical manner, and having empathy goes a long way in ensuring that the ethics are maintained or upheld. Now, ethics in biomedical research is very similar to ethics in clinical practice, even very similar to, like, for example, when you students, you go to the clinic, I mean, in the wards, in the clinical wards, and you want to, for your clinical classes, you want to take the history and examine a patient. So when you approach that patient, you know that you may not be benefiting, your approach to that patient may not be benefiting that patient. The patient is under a particular doctor, a particular team, particular specialty, and that specialty is who is trying to help the patient recover from their illness. But when you go there, you are only going there so that you can take the history and examine that patient for your class, so that you can learn. So you still have to convince that patient, you have to take permission from that patient to be a part of your class. So in order for you to take history or you examine that patient, you can't force yourself onto that patient. Even, for example, if the patient has acute appendicitis, you can't forcibly palpate the abdomen when it's very tender. You have to be ethical. So unless the patient gives you permission to participate in your clinical classes, you will not be proceeding. You will try and ask the next patient. The similar way, when you are performing or when you're going out to perform research, you need to make sure that ethics are upheld in a similar way. So any participant that is included in your study, we must make sure that or as an investigator, I must make sure that the ethics are not violated. The participants' rights are not violated, be it a human, be it an animal. So this is a very nice saying that by Aristotle, educating the mind without educating the heart is no education at all. So ethics is, of course, not just restricted to biomedical research or not just restricted to medicine. It's something that should be part and parcel of society. So how we inculcate ethics is right from our homes, so from family, from school, and this is how we can carry it into the field of biomedical research. Why do we need these ethics? Now, this is a very famous photo. Anybody can identify what this is?
So, torture cell or a jail?
True, true. Very good. So I said famous. Actually, maybe the better term is infamous, because this is from the Auschwitz gas chamber. This is in the Nazi camps where Nazis used to forcibly experiment on other human beings, Jews mainly, in concentration camps where they were subjected to nerve gases to determine how deadly the nerve gases are, or at what doses what effects are produced. So probably these scientists might also have had their research question. They might have had their objectives and methodologies as part of the protocol. But in going about executing that particular protocol, they did not pay attention or deliberately neglected the participants' rights. Right. So did the participants consent or volunteer to be a part of this study? No. So in as and when these kind of incidents, these kind of experiments, immoral, illegal experiments came to light, there were conventions to try and guidelines were formed to try and correct these and put these into the right, have these experiments conducted in the right manner. So similarly, there have been other examples in the past century. For example, this is the Hillsbrook School. It was meant to be a school for mentally challenged children as well as adults. But there in the Hillsbrook school, where the mentally challenged children were left behind, they were intentionally by certain scientists, certain medical scientists rather, intentionally infected with hepatitis. And maybe as a result of those experiments, we got a lot of information, including the difference between the mode of transmission of hepatitis A and hepatitis B. We got information about the effectiveness of immunoglobulin therapy in hepatitis A. So scientific advances made were true, cannot be dismissed. But at what cost? These were experiments conducted on children who could not consent. First of all, children cannot consent to their own on their own. They need to be under the care of a proper guardian, a parent if available. But experimenting on children, especially those who are mentally challenged, who've been abandoned, so to say, in a school, was probably one of the worst violations of ethics in biomedical research history. And one more example is the Tuskegee experiment that was conducted on African Americans in the US. So even as late as in the late 1970s, these experiments were being done where syphilis infected African Americans were denied treatment just in order to be able to find out how the infection progresses. So we may never have got to know how tertiary syphilis manifests unless these kind of unethical experiments were conducted. So maybe scientific advances, yes, but at what cost? So patients who could have been treated, who could have been cured right in primary syphilis stage, ended up succumbing, and generations together suffered. So there have been several incidents like this where experiments have been conducted and participants have not been treated as participants, rather they've been treated as human guinea pigs, which we must, so to say, guinea pigs also have their own way to be treated even in these animal experiments. So we can't be dismissing the rights of any participant, be it human or animals. So like I said, every time these kind of incidents came to light, we had conventions and guidelines coming out, starting from the Nuremberg Code right back in 1947, the Helsinki Declaration 1963, the Belmont Report 1997, the International Council on Harmonization of Research in 1997 again, and even there was an update in 2016. We have our own guidelines as well from the Indian Council of Medical Research. The ICMR has its own good clinical practice guidelines on how to go about when performing research, making sure that there is no violation of ethics. So these guidelines go a long way in helping us to make sure that we are not on the wrong path. So we may have set out to achieve something in science, but it cannot come at the cost of human values. Okay, so there are certain principles. There are several principles in with regards to ethics in biomedical research, but the main ones which have been quoted as core principles by the scientists, by the Childress and Buccaneer, are these four. Autonomy, that means the participant should be able to decide. It should be voluntary. It should not be enforced. It should not be under duress. If the participant wishes to participate in the study, then they may. Otherwise, you cannot have them forced. For example, how the Nazis forced Jews into all those experiments in the concentration camps. Non-maleficence and beneficence, these go hand in hand. Non-maleficence means do not harm. Do not harm, for example, in patients with the in the Hillsbrook example, where the mentally challenged children were given intentionally given hepatitis A infection, that is harming. So we cannot have that being done when there is no chance, no, where there was no risk of developing hepatitis A infection. It was intentionally done. So non-maleficence means do not harm. Beneficence means do good. Okay, so that would be the Tuskegee study where syphilis patients were denied treatment. So in denying them treatment, we have done them harm. So these go hand in hand. Do not harm. Instead, do good when you can. Intentionally, do not harm them. Okay, there may be some harm involved in your experiment. There may be, for example, a clinical trial which is exploring the role of a new drug. There may be some side effects which are not yet known. So these have to be explained to the participant before they can be enrolled. They must be fully aware that there may be a risk, but they are aware of the risk and yet wish to participate. And maintaining dignity of the participants, justice. So, for example, like the Tuskegee study was conducted only it was a racially profiled study. It was conducted only in African Americans. So that again goes against the principle of justice. And it's not limited to these 4 core principles. There are several others, and you would realize that all of these are common to when tomorrow you become doctors and you're practicing and you are interacting with patients, you will be making use of all these principles even in your clinical practice. So it's not that these are exclusive to research ethics or in the field of research. They're common to even clinical practice. So these are some of the qualities that need to be inculcated by all of you before you before you graduate. I'm sure most of you already have them, but these are to be kept in mind and never to be abandoned. Okay, so when we are thinking from the researchers, from the participants' point of view, how are you recruiting them? You need to make sure that it is voluntary and it's not binding. It's not a contract. They can, if they feel they are not comfortable, they had initially enrolled, but they're no longer comfortable in continuing, they can opt out of it anytime they want. So it cannot be a forced participation. It cannot be that, okay, the participant agreed at the beginning, so that means he cannot go out of the study until the study is complete. That kind of, you cannot restrict the participants' freedom in that manner. The risks, as I said, risks and benefits have to be explained. Actually, this benefits is why all this ethical issue comes up. Because, like I said, when you go and examine a patient in the ward, you may not be doing anything directly beneficial to them. In the same way, many of these experiments, or many of these trials rather, many of these research projects may not be directly beneficial to the participants, but instead, after the analysis of the results from these trials, there may be benefit to the larger society. So when it is not going to be directly beneficial to the particular participant, it may be a difficult uh, balance between doing the project and not infringing on the patient's right. So that is why it is very important to make sure that you discuss in detail what the project is, how the patients may be harmed if there is any risk, or how the society may benefit from their participation, even if they directly do not. Compensations. Now, certain trials may offer compensation, but in what form? The compensation is mainly to be in the form of reimbursements. So if they've had to travel for in order to come and participate in the study, or if they've had to lose some of their work time, that amount of money can be given as compensation in case there is any, let us say, a disability or a disease or a hospitalization, or worst case scenario, even death in a particular clinical trial. In that case also there can be compensation, but it cannot be a paid honorarium to participate. So the participant cannot be made to think that he'll be getting money and therefore the person must participate. So it should not be in the form of a coercion. It should rather be in the form of compensation. So, or any procedures that the person may have to undergo in the form of investigations, or if in case the person has to undergo a procedure, a surgical procedure, whatever it is, that needs to be clearly explained to the participant. Or in case the participant needs to take drugs which which may be standard drugs or which may not be. If it's a blinded trial, then even the participant may not know. So that needs to be explained, that they be given a drug which they will not be aware of, whether it is a standard drug or a placebo or a new drug. And in case at any point of the trial or study they have any grievances, there needs to be a mechanism for them to be able to address. So they need to be given contact numbers or details to whom they can report to in case they have any issues with regards to them being part of the study. These mechanisms need to be put in place even before the studies begin. So Nankumar sir said, as Nankumar sir described, there are these vulnerable groups. So vulnerable groups are participants who may not be able to make their own decisions, take their own decisions. They may not be able to defend their own rights. So one example I did give was children. Can you think of other groups who may not be able to defend themselves in case they're being forced into to participate into a research? Who could be vulnerable? Who could be
Retarded adults.
good? Just like
Elderly.
good. Yeah. Anyone else? So, like in the concentration camps of Germany, prisoners. So prisoners could be anyway. They have very limited rights. So even those rights could be taken away without them being able to appropriately express uh, that kind of violation of their rights. So, like you said, children, socioeconomically deprived, so poorer sections of the society may not be educated enough, may not be able to comprehend what is being done to them, or what is being asked of them, and how their rights may be getting violated. So, these are just a few examples. There could be, it's not restricted to these. Anyone who may be whose rights may be already compromised are already vulnerable. So, like I said, if I wanted to talk 45 minutes or 1 hour, you being students, I am sort of coercing. I am using my rights, or they're not rights, but my power or my position as a teacher. I'm infringing on your rights and forcing you to listen, maybe when you don't want to. So like that, whenever there's a person whose powers are limited, who's not able to express or defend their own rights, they become vulnerable. And such vulnerable groups, you need to be extra careful. It's not that you need to exclude them from the study, but you need to make sure that their rights are respected, and there's no infringement in their uh, upon their rights. So one of the ways you can do that is making sure that an informed consent is distributed, and you have a copy with you and one copy with them with their signatures on it. So the patient should know exactly what this study is, what is the purpose, why they are being invited to. So it's like an invitation. They're being invited to participate. So only if they accept the invitation, they enter the study, they get enrolled. Otherwise, they have every right to not participate or even drop out once having accepted it. So having an informed consent form with a patient information sheet. So PIS stands for patient information sheet. That should have details of all these things, so that as common language as possible. So even a layman should be able to understand what exactly is going on, and if there are any risks involved, if there is any chance of benefits, if there is any reimbursement involved, in case the person has any grievances, whom can they contact, or how do they express those grievances? All these mechanisms need to be put in place to make sure that the participants' rights are not violated. Okay. So informed consent, like I said, language should be as common language as possible. It should be in the colloquial language also. So if, for example, the study is being conducted in Bangalore, it should be that there should be a consent form in Kannada also, in the local language. So the person should be explained, as well as should be able to read in his or her own language. In case the person is illiterate and not able to read, then the person should be explained in the language that they understand, and the thumb impression along with the signature of witnesses need to be taken. You need to make sure that you are you are also safe. So in case you do enroll a participant, and tomorrow the participant goes to the ethics committee and says, no, I was forced to join the study when I didn't want to, then you will be it'd be difficult to defend yourself unless you have that informed consent form which is signed. Even if the patient is illiterate or the participant is illiterate, you can get a thumb impression with the signature of unbiased witnesses. Okay, so it should be detailed and should have signatures, like I said. This is an example of one of the universities informed consent form. So it would detail out what the study is, what the participant may need to be undergoing, as well as having a place for the signature and along with the date. In case the person is illiterate, then it would be thumb impressions along with signatures of witnesses, unbiased witnesses. Okay, so how else can you safeguard the patient's rights? So it's between you and the participant, it's fine, but then we need to put in additional safeguards so that there is no chance of any violation, ethical violations. So we have, of course, the institutional ethical committee. Now it is mandatory that absolutely every study should pass through the institutional ethical committee, or in the West, it's called an institutional review board. So ethical clearance needs to be obtained for any research project. So whether it is prospective, retrospective, or cross-sectional, it needs to pass to the ethical committee. The ethical committee institutional ethical committee will have members, will have doctors as well as legal consultants then from outside of the particular institute also. So this helps to analyze what exactly the protocol is, whatever the study protocol is. In case there is any chances from their perspective, they be able to bring out what issues, what ethical issues may occur as the study is starts, as the study is underway. So apart from that, at various levels, just examples I've given. Health Ministry Screening Committee is for any international research that is happening. Then there needs to be a coordination between the centers from the different countries via this health HMSC, Health Ministry's Screening Committee. Data Safety Monitoring Board. This is for clinical trials, and there are other committees also, but the Institutional Ethical Committee is the main committee which will be supervising the study, and they have the right at any point to intervene in case they need, in case they feel it's required. They may ask for the data to be produced, and at regular intervals, reports need to be, the studies, the research reports, the project reports needs to be handed out to the ethical committee in order to make sure that there's no violations of ethics. This is another example of what you can do to make sure that you're not going to be violating any ethical or not performing any unethical practices during research. This is certification of good clinical practice. This is something that a number of courses that are available even online. It could involve just reading of material and then passing exams, or it could involve video lectures. So this is in fact mandatory for clinical trials. Good clinical practices certification is mandatory for clinical trials, but even for non-clinical trials, having this kind of a certificate goes a long way in ensuring that, in displaying that yes, you know what the proper ethics are, and that you will not be violating any ethical principles as you conduct the research. So, like I said, non-interventional or retrospective, whatever be the design, it is best to pass, because even though there is a little bit of it is a little bit of a gray zone with respect to retrospective studies, whether the ethical clearance is required or not, but it's always best to be safe. So passing it through the ethical committee, it may get expedited, but always better to pass any research project or protocol through the ethical committee. So yeah, and one last word on publication ethics. So ethics cannot be restricted only to research. The project itself, the project may finish, and then you'll be wanting to write it up and publish it. So ethical practice should continue even into publications. So I've just mentioned these three terms: plagiarism, fabrication, falsification. You need to also disclose any funding. You need to disclose conflicts of interest. So all these are also there. So publication ethics is again another subtopic which we can focus on in the later weeks. So, but for today, we'll restrict to only research ethics specifically. And just before I finish, I just want to highlight, this is how now that we're glad that all of you have been able to log in today and attend this session. And as Nankumar sir emphasized, it should be more interactive, and you should not hesitate. This is not going to be a session where we're going to give you marks or maybe deduct your marks from your your priority subjects. But this is a sensitization session, so we want all of you. There's a lot of potential which we want it to get fulfilled, and that's the only purpose of this conducting these sessions. So any of you who do wish to start new projects and are not aware of exactly what the workflow is, how the workflow is, how you're supposed to go about, I just display the slide where you can have a decide on a guide. If you have a topic yourself, then you can take it to your the relevant guide, this particular specialty or particular department, and approach a particular faculty. All faculty from all different departments are always willing to guide students in their quest to perform research projects. So, or in case you're having trouble coming up with a research question, you can always come to the Division of Research and Patents, and we'll be helping out with that. And then after preparing a brief synopsis, you can submit it to the Division of Research and Patents, and to obtain your unique research number. So your unique research ID is important. You have to approach the DRP first, get the DRP number, and then you may approach the student research committee, probably after discussing the statistician regarding sample size. Okay, so the first thing you should be doing is approaching the DRP once you have a topic in mind and you have decided on a guide. And beyond that, of course, this is what we had discussed. The ethical clearance needs to be obtained once the protocol has been finalized, and only once the ethical committee clears it, the ethical review committee clears the topic for a particular valid period of time, then you can proceed with data collection. Okay. So this was just a brief session. It meant to just act as a sensitization course. So we will be trying to bring you a little more hands-on experience with more examples and discussion sessions, where maybe we can also have some face-to-face sessions also, maybe in limited numbers. We can plan out for those in the next coming few weeks. If anyone has any questions, any doubts, we'd be very happy to take them now.
Excuse me, sir.
Yeah.
So while discussing, Chidambaramurthy sir told about the importance of a mathematician and a statistician. No, I couldn't understand the points of what he meant.
Yeah. What was that? Was it Ullas?
Yes.
Yeah. No, the thing is, as we discussed, once we collect a set of information, what method we use to analyze and represent the data. For example, if you take the body mass index of 100 people, and you can't say like 100 person name and write the body mass index. There is a way to represent it for easily what we say interpretable manner. So for that we have to do a statistics analysis. Okay, so when we have to do a statistics, there are ways and means in which you have to collect the information. It is not a one parameter you will be studying. You'll be studying multiple parameters. So in order to get those things, and suppose if you want to do a particular study, how many subjects or how many experiment you need to do, all those things, we call it a sample size selection and calculation. Those things we need to take the support of statistician. As in medical doctor or as other researcher, we may not be able to decide on that. For that, most of the student, especially when they are doing their dissertation and project, they complete all the work and finally they try to meet the statistician. So that time he may say, like, if we have these parameter, we can analyze better. And those are the things which we need to consider. So probably when we in future we talk about statistical methods and tools, you'll get a better picture. Is that clear to you, Ulas? Does it answer your question?
Yes, sir.
Okay. So if there any other question, please send the type question, or anything in the chat box we can just address. Any other quick question before we wind up today's session?
You said about some an ethical certificate or something. Is it wise that we go about it now? I think some certificate, some training we have to do. Is it wise to go about it now so that we'll be like pre-prepared or something?
Hi, thanks for the question, Varun. No? So, no, what I meant was for your protocol, you will get an ethical clearance from the ethics review committee, our institutional ethical review committee. So, for that particular protocol that you submitted, after they have deliberated, they may point out or suggest corrections or pass it as it is. But unless that clearance is there, you will not be. It will not be right to proceed. Regarding that good clinical practice certificate, unless you're it's not a mandatory certificate for non-interventional trials. So unless you're planning for interventional trials, it's not required because it will have validity also. So maybe when you do decide to participate in a clinical trial with intervention, randomized clinical trial with intervention, where you would have an arm which will receive placebo, arm of participants who receive placebo, and another arm who receive a novel drug. In that case, it'd be better that it will be required. Most clinical trial agencies which do perform clinical trials do mandate that the GCP certificate be there, but it has a limited validity. So unless it's on the horizon where you're about to going to participate now, it wouldn't be required. Not for most of the projects that we do with students undergraduate level are non-interventional type, for which you don't need a GCP certification.
Okay, sir.
Any other question?
I think we'll call it a day.
Yeah.
And I would like to express my thanks to the college management who have been kind enough to create this division of research, the principal and dean, the vice principal, Dr. Shalini ma'am, the registrars, and the team at Division of Research and Patents who have been extremely helpful in organizing, and the coordinator for today's program, Dr. Akshay Rao, for his relentless effort in ensuring the undergraduate research training happens on a regular basis. And this is the first of the series that we are planning. And most importantly, the students who have shown keen interest, and I would certainly want you to participate in all the future trainings as well, so that we can help you to undertake quality and impactful research for the years to come. So thanks a lot everyone for joining us. Have a nice and safe day. Thanks a lot. Thank you.
Just a reminder, students, please submit your pre-test answers and encourage all your friends to submit it also. We need those email IDs for us to be sending out the certificates later on for participation. The post-test questions will be sent out later. Thank you very much, Nanda sir, and thank you, Chindamurthy sir.
Thanks a lot.
Thank you.
Tanuja ma'am also. Thank you to all our students who participated today, and we'll be seeing you soon.